Welcome back to Bio 111. This is chapter 16, section 3. We'll be looking at eukaryotic epigenetic gene regulation. A few things to recall about eukaryotic gene expression. It's more complicated than in prokaryotes. We just talked about prokaryotes. Transcription is physically separated from translation. Transcription happens inside of the nucleus. Translation happens inside of the cytoplasm. Remember, transcription is where you take the information from DNA and you transcribe it into RNA. And translation is where you take the information in the RNA you just created and then translate it into protein. The process of going from DNA to RNA to protein is much more complex in eukaryotes, meaning there are more steps. Because there are more steps, there are more places where you can control this flow of information. So regulation can occur at many levels. The first level begins with access to DNA. We call that access, that control of access to DNA, we call it epigenetic regulation. This is relatively new in terms of other forms of understanding of regulation. And it occurs before transcription begins. We also have these proteins we call transcription factors that bind to DNA and help control the transcription of that segment of DNA into RNA. A few things to remind you about the human genome, our genome. We've got about 20,000 genes. It's likely that more will be discovered, but right now we know of 20,000 genes. We have 23 chromosomes, and that means that of these 20,000 genes, they're spread across these different chromosomes. All right? If you recall, when we talked about DNA, eukaryotic DNA, it's really long and has to be compacted. We do this by wrapping our DNA around little spools, like thread on a spool, we call histones. Now, you cannot access, just think about this visually. If you've got the DNA wrapped around this histone molecule, like a spool, you can't access it. You've got to be able to physically access it to read it to make RNA from it. The molecules, the polymerases that must attach to DNA cannot do it when they're wrapped around, when the DNA is wrapped around these histones. So expressed genes must be unwound and made available to polymerases. So that's where this control comes in. All right, this is a little cartoon about showing DNA being wound around a histone molecule. You have a picture, an electron microscope image to the right, image B, that shows what this looks like inside of an electron microscope. Gives you an idea why scientists call this beads on a string. It really looks like little beads attached to little strings. All right, so if you look at this, when nucleosomes are placed closely together, the mechanisms needed to attach to DNA cannot do so. We have this little side note that methylation, the addition of a methyl group to the DNA, and histones, causes nucleosomes to pack tightly together. So when a methyl group is added to either the DNA or the histone protein, the DNA is more compacted, right? That means that we cannot transcribe that DNA, all right? When the nucleosomes are spaced far apart, the DNA is exposed. It's made available to transcription factors and polymerases. All right, so histone acetylation results in loose packing of nucleosomes. So acetylation That's when we are adding an acetyl group to the histone protein. All right. This spacing out allows for the DNA to be accessible or accessed by these factors and by the polymerases. All right, so methylation in general suppresses gene expression, and acetylation increases gene expression. All right, so that's the basics of epigenetics. Right, we're modifying the gene, not modifying the genes, we're adding tags to the DNA and to these proteins that they're wrapped around. Right, we're not changing. We are not changing the sequence of the base pairs in the DNA. We're not changing your DNA. We're changing how it is read and how it is folded. All right. With chemical tags. So chemical tags are added to histones in DNA. We have phosphate, methyl groups, acetyl groups. They serve as tags. They're not permanent. They can be added. They can be removed. All right? And they act as signals to tell histones if a region of a chromosome should be open or closed. All right? This is the basics. Like I said, this is the basics of epigenetics. Now, epigenetics, we like to talk about what words, where they come from. That means around genetics, all right? We're looking at how we're controlling kind of on top of the DNA. Temporary changes to nuclear proteins and DNA that do not alter nucleotide sequence, but do alter gene expression. All right. Just looking at this as a little bit of a cartoon showing sections of the DNA that have been methylated, they're packed, sections of the DNA that have been acetylated, and they're accessible, they're loose and accessible. Right. All right, so that is section 3. The next section, section 4, we're talking about eukaryotic transcription and gene regulation.